Students are often unsure of where to start with their revision. What is often overlooked is familiarising themselves with the specification that they are studying. What is the content? What knowledge and understanding do you need? How will it be assessed? Is there anything in the specification that will help you in your exam success? So today we're going to look at the AQA Combined Science course, the Trilogy course. We're going to take a look at the specification at a glance, look into the required pracs and have a dig into the specification to see if there's anything in there that will be helpful to you in your preparation. Remember, if you want to pause the video at any time, please feel free to, or go back if there's something that you've missed that you'd like to go through again, and that will help you get the most out of this presentation. So let's have a look at the specification at a glance. As a combined science course, it consists of biology, chemistry and physics, and you can see the different components that each of those are made up of. You will have your own strengths and weaknesses in those areas, and familiarise yourself with all the names that are here. So for instance, bioenergetics at first sight might be a bit confusing, but that's referring to the work that you've done on enzyme activity. If you're at the start of year 9 or year 10 or year 12 or year 11, you're going to be at different points in this course. So a good technique for you to do is review the work that you've already studied, and that will make good building blocks for you moving forwards. I'll take a minute here just to pause the video and review what you have already studied, where your areas of development are and where your strengths are. And it's a good starting point to have an overview of where you are in this course. Looking at the required practicals, there are 7 for the biology, and you can see here, I've taken a lot of this information from the specification, that there's one on light microscopes, which will also involve animal cells, animal and plant cells and magnification, one on osmosis, looking at sugar and salt solutions and how they have an effect upon plant cells. Most often used is potato, but don't be thrown out if they suddenly use carrot or something like that. The same principles apply. Food tests of carbohydrate, lipids and proteins. You should hopefully remember that Benedict's reagent is used for carbohydrates, that iodine solution is used to identify starch, and the Buret's reagent is used for proteins. You also look at investigations on how a range of pHs will affect the rate of reaction for the enzyme amylase. Light intensity and rate of photosynthesis, so you should have an idea about that already. Reaction times and developing an experiment to investigate that, and measuring the population size of a community or a species within a habitat, and looking at how that distribution is affected by a factor in the area. So that's the biology, 7 practicals there. And then if we go on to the required practical for chemistry, there's 6 practicals here. We're looking at this preparation of a dry sample of a soluble salt. Most often used there is copper sulfate crystals, making that from copper oxide and sulfuric acid, reacting those two together and forming that. But other forms might be used. Looking at electrolysis in the second practical, and then we're looking at this change in temperature. So in terms of these different reactions here, and then concentration rates of reaction for the 4th practical, paper chromatography and analysis of pure water samples there for the 6th one. In the physics, we have the 8 practicals here: specific heat capacity, resistance, especially through the length of a wire kept at a constant temperature, and then through combinations of resistance in series and parallel. Looking at this IV characteristics for filament lamp diode and resistor at constant temperature. Ways of determining the densities of regular and irregular solid objects and liquids. Relationship between force and spring. And then looking at acceleration. Frequency and wavelength and speeds of ripple tank. And then the Leslie cube often is the one that's used here in terms of the investigation for infrared radiation. So that's 7 for biology, 6 for chemistry and 8 for physics. 15% of your papers will be on the required prac, so this is an area that you can definitely get ahead of the game in, because these are definitely going to come up in some form. You won't know which ones for sure, but when you do one, they can draw on these in the examinations. So take a minute to go back through those, familiarise yourself with them, and make sure you're aware of which ones you've done and which ones you need to revise. So we're going to have a dive into the specification here. So I've put the link here in the presentation. I'll also put it in the description. Things that you need to familiarise yourself with really are the periodic table, the physics equations, which you will not have a copy of the equation sheet in the exam. It's something you can get ahead of the game with and learn them. We'll have a quick look at the assessment objectives included in the specification, so you get a bit more of an idea about what's being assessed and how much the weightings are for those. So if I click on the link, that will hopefully take us to the specification here. Important to check that it is the right specification. You've got the page here, which is the contents page, and very handily you can click on that and you can see all the different areas of content. So for instance, if you were looking at cell biology, for instance, and cell biology was causing you some consternation, then you could go and have a look at exactly the content as it's laid out here, so that you can make sure that you have an understanding of that content. Read through these points, and then that will allow you to get some idea about where the content's going with that, and that'll give you ideas about skill development. So the ideas of centi, milli, micro, nano, and understanding what those prefixes means there, for instance. And you can see some of this is actually providing you a checklist of things that you need to be familiar with. Okay, if I just go back to page 3 here, which is the contents page. Another really important part to have a look at is this key ideas. So you've got these key ideas in biology here. So this applies to all the study of biology and is something that I'd familiarise myself with, just giving you an overview of those overarching ideas. And what you can see here on page 3 is that there's key ideas for biology, for chemistry, and for physics. Right, quick look here at the assessment weightings and the assessment objectives. If we look at these, we've got these 3 outcomes: assessment outcome 1, 2, and 3, and you can see the weighting is 40, 40, and 20 for assessment outcome 3 here, objective. So you can see that's about knowledge and understanding. It's also about of scientific ideas, techniques, and procedures. But then we've got inquiry techniques and as well. So that is the difference between the first and the second one there. And if we look at this third one here, we can see that we have this interpret and evaluate, make judgments, and draw conclusions. And this is going to allow you to develop and improve experimental procedures as well. So that's that idea of that experimental work as well. Okay, if we go back to page 3 again, we can go down here, we can have a look at the periodic table on here where we've got there, so you can familiarise yourself with the periodic table, print out a copy, make sure you're aware of all those different groups, group 1, group 2 transitions. Can I turn this page, rotate clockwise, there we go. So we've got it nice and clearly there in front of you, and we can see that. And then we can move that back and have a look at the equations now as well. So appendix here on page 195 of the specification, the physics equations that you need to be aware of. You'll see that here, for instance, we've got equation number 7. It's got HT, which is only required for higher tier, and we can see here with these equations here at the end here, again higher tier and higher tier for these ones here. All right, so those are the equations that you need to familiarise yourself with. That's where they're found, and that should provide you with a good source in which to do that. Okay, so I think that covers everything I'd like to go through with you. I hope you found that useful. Please let me know, like, subscribe, and let us know what you think of that presentation.